So thank you, Robin. It's a pleasure to be here. I just want to let you know that it is 3 o'clock in the morning in Oregon. So sorry if I'm a little jet lagged. I was thinking, yesterday and today, the very first mycological meeting I went to was the Fungal Holomorph Conference in Newport, Oregon, years ago that John and Don Reynolds organized. I know Don is here. And so this has kind of come full circle for me. It's pretty interesting. And that had a big impact on me as a young mycologist and developing in my career and field. And I hope that those students who are here, this conference has a similar effect. So I want to talk about, or I was asked to talk about this project that we've started to sequence 1,000 fungal genomes. And I don't want you to really focus too much on the absolute number there. It's really more to kind of capture your imagination of thinking big about sequencing genomes and to think about it in a systematic way. And what I want to emphasize, or get around to emphasizing, is that really we need to... This has the mouse or no? Okay. No. So I want to emphasize the role that taxonomy has to play in picking what genome. So this was touched on by Conrad and a few others. So this concept of one fungus, knowing this one species, what genome would you pick? So as an overview, this 1,000 Fungal Genome Project is really a second project that's funded is not the right word, it's happening at the Joint Genome Institute, which is a unit of the Department of Energy. And the Joint Genome Institute, if you're not familiar with it, they're focused on the development of biological solutions using microorganisms around the carbon cycle and carbon sequestration, development of alternative biofuels, and bioremediation of soils, contaminated soils. The first project that we had, and I'll tell you a little bit about, was associated with AFTAL, and it's the Early Diverging Fungi, CSP, for a community sequencing program. And then the second one that developed from it is this 1,000 Fungal Genome Project. And these are some of the individuals who are involved with that as PIs or co-conspirators, so to speak. Our starting point was our understanding, our hypotheses of phylogenetic relationships of kingdom fungi after the first AFTAL project. And this is a cartoon of that showing the major lineages of phyla and subphyla, where you see the little circles with the blue circles with squiggles. Those are to denote zoosporic lineages, things that have been considered chytrids or chytridiomycetes at some time. The rest of these early diverging fungi and the basal lineages that don't have zoospores are things that have been considered zygomycetes or zygomycota at one time or another. And then you have the dikarya with the ascomycota and basidiomycota. Where you see the names of species names are those that we've sequenced as part of the early diverging fungi CSP. And there's 5 of them that are complete, and there are an additional 5 that are in progress, and that's denoted by the superscripts 1 and 2 that you see. I want to highlight here, and do I use this to highlight on the screen so that people can see it? And how do I do that? I'm not... Oh. Okay, there we go. So I want to highlight a couple things. First, the Monoblepharidales, where we've sequenced the species of Gonadopodia. There are 2 placements of this taxon. One that places it within the chytridiomycota, and a separate one from a different study that placed it as possibly one of the earliest diverging lineages of the kingdom fungi. Now, the Monoblepharidales are unique amongst the chytrids in that they have oogamous sexual reproduction. So a non-motile egg that's fertilized by a motile male gamete, an antherozoid. So it's unique amongst the fungi. And then I'll show you a second example within the basidiomycota, where we've always had a difficult time resolving the relationship of these 3 subphyla and the effect of taxon sampling at a genome scale with respect to resolving some long-standing problematic issues. So on this first one, looking at the chytridiomycetes, chytridiomycota. So this sampling here, there are 3 genomes that were sequenced amongst the early diverging fungi. And this work really comes out of Mary Burby's lab at the University of British Columbia, who's one of the PIs on this CSP. And the 3 that have been sequenced are Gonatopodia, which is a member of the Monoblepharidales, and then 2 additional zygomycetes, or zygomycota in this group, a Comansia from the Caxellomycotina and a Conidioblis from the Entomopteromycotina. These are the size of their genomes, excuse me, and these are the number of predicted gene models or proteins from those genomes. A couple things I want to point out is first that Gonatopodia is well placed, strongly placed within the core chytrid clade. So that earlier result of it potentially being a basal or the earliest diverging lineage of the kingdom doesn't seem to hold up in these phylogenetic or these genome scale phylogenetic analyses. This also creates a bit of a problem for taxonomy in this group, in that Pyromyces of the Neocalamastogales is recognized as its own phylum, the Neocalamastogomycota. Superfragicalistic expialidocious. But so this, I think, would make a good case of recognizing this entire group as a chytridiomycota and understanding that some of these morphologies, like we so often see, are odd apomorphic morphologies within a particular group. The other thing to point out is there are still 2 core zygomycota clades, the Mucoromycotina, and then, which we have here is Zygomycota 1, and then the second clade of Entomopteromycotina and Caxellomycotina, or Zygo 2. Where you see these 2 asterisks represent clades or nodes that are unstable in these analyses. Those taxa, in particular, Allomyces from the Blastocladiomycota and the Zygomycota group 2, those move around in these analyses, excuse me, depending on which sets of genes you look at. And I think part of this is explained by inadequate taxon sampling. And that's going to be a problem in genomic studies for a while. And as an example of that, this is work that included work out of Kathy Amy's lab, who did the genomic work, and Dave McLaughlin, who did the ultrastructure work, the TEM work on septum morphology. And this involves Wallemia. If you know Wallemia, it's detected as a household indoor mold associated with dust. It's also one of the osmophilic fungi and can be isolated from environments like molasses and such. Its septal pore structure looks a lot like members of the Tremolales. There's a dolly-pore septum. There's a septal pore cap that's derived from the endoplasmic reticulum that has these saculator finger-like projections you see here. This is quite similar to that of Cryptococcus. Wallemia in these analyses is placed as the earliest diverging lineage of the Agaricomycotina, so consistent with that morphology. But what I really want to highlight here is this node that unites the Agaricomycotina with Eucelaginomycotina. The 3 subphyla of the Basidiomycota have been problematic in their resolution, even with genome scale data. Depending on what you look at, you can get different resolutions, relationships between those 3 taxa. However, here we have a very consistent signal, and in addition to that, you see these 3 numbers, 84, 96, and 100. These represent the bootstrap values with increased removal of hypervariable sites. So what we did is we bend those sites into 8 categories of variability, with 1 being the most conservative, 8 being the most variable. This is with all the data, 84. This is removal of the fastest site, and this is removal of the fast 2 sites, categories of sites. So it's thousands of amino acid positions. And by doing that, you see you arrive at stability of that node. And this is not only true for this node, but we see it in other nodes of the tree. So one of the take-home messages here with genome scale data is even though you can sample all the data, doesn't mean you should necessarily analyze all the data. So this is our current sampling of the kingdom fungi. Now, when we look across the kingdom, where you see check marks, there's some genomic representation. Where you see the CSP, the EDF CSP, that was sequenced as part of that effort. There are a few that are still problematic. Opidium, which is a chytrid that's a parasite, root parasite of Brassicaceae and other things. And here are some other obligate pathogens or obligate symbionts of the Zoopagomycotina and things like the rosellids or the Cryptomycota. So we're getting good coverage, but our coverage within these classes is still quite poor. And that's what F1000 is focused on, really expanding, going deeper into coverage. And there's a number of goals here. The first is to facilitate the sequencing of 2 genomes from every family level clade of fungus. So there's about 560, 65 families of fungi. Several of those already have representation. We're focused on using these data to resolve the deep and problematic nodes, unresolved nodes of kingdom fungi. We're very interested in using these data to provide a more complete overview of fungal metabolism, especially in the context of the mission of the Joint Genome Institute and the Department of Energy. And importantly, to begin the development of reference data for metagenomics. As John mentioned, 1,000 is not going to be the answer, but it is a starting point. So this is a phylogenetic tree, a cartoon of the kingdom fungi. Here you see the major groups. I've simplified them, the early diverging lineages, the 3 main subphyla of the Basidiomycota, and the 3 main subphyla of the Ascomycota. And these are the classes or subphyla that occur in those. This first set of bar graphs are the absolute number of families that occur in those in the current classification. Where you see blue, those represent the numbers that have genomic representatives. Where you see red, that is the focus of Tier 1 sampling of F1000, or the first year of sampling. And where you see green are the remaining, the unsampled taxa that still need to be sampled. On the right, these histograms show that same data but relative numbers. So you can see for some groups, like the Tephrinomycotina, there's good representation of at least 1 or more genomes from all of the families associated with those groups. And you can also see that for some groups, like the Puccinomycotina, there are major gaps in our phylogenetic coverage considering families. Now, this is just showing this Tier 1 sampling in a little more detail. We've selected 100 species, 100 isolates for the first year. And we've intentionally chosen what we think or what we naively consider to be low-hanging fruit. And the reason we did this, if I go back one, if you look at the Sordaromycetes, for example, here's the blue, what's already been sequenced. Here's the red, what we're proposing. So after this first year, theoretically, we should have greater than 50% coverage of all of the families of the Sordaromycetes. Same thing true for the Dothidiomycetes and the Uroshiomycetes to greater and lesser extents, and the Agaricomycetes, excuse me. So here's that first tier sampling, 25 Dothidiomycetes from 25 families, 19 Sordaromycetes from 19 families, and so on. You can read that. What I want to point out over here is the role of culture collections. Culture collections are front and center in genome studies. You're able to work with vouchered material. You're able to work, in the case of CBS, with a very active barcoding project where if the correct name isn't on there, there's a path to a more correct name. But in addition to CBS, which has a lead role in this, NRRL at Peoria, Illinois, USDA collection, the Fungal Genetics Stock Center, the Center for Forest Mycological Research in Madison, Wisconsin, which has a fantastic collection of mainly wood-rotting fungi. And then here, this endophyte collection, this is the Arnold endophyte collection at the University of Arizona that Betsy Arnold has put together. So Tier 2 and beyond, and this is a 5-year project. There's a number of things to think about. And I think these can lead to discussion or questions. And one is we want to continue focused on this unsampled subordinal clades, family-level clades. And I want to emphasize that families are guidelines. We have a good understanding of the monophyly of the major classes and subphyla of fungi. We have a good understanding of the monophyly of many orders, if not most orders. Not so much at families. So when you think about this subordinal sampling, and I say families, it's really as a guideline for sampling monophyletic subordinal clades. We need to coordinate with other genome initiatives, including the Chinese Fungal Genome Initiative, the Broad Institute, which is in the US, the JGI Genoscope in France, and many others. The most difficult one to coordinate with are all the small private labs that are now doing genome sequencing on their own. This is becoming easier and easier. So our ability, we need a mechanism, a better mechanism than we have now with respect to a clearinghouse, if you will, or a way to communicate this more accurately. Most importantly, perhaps, is we need to expand the role and engage the community of mycologists, including taxonomists, ecologists, and culture collections, to participate in this. So what will happen here is that if we accurately communicate where the gaps are, and you can fill one of those gaps, your genome will get sequenced for free. You'll have access to that data. The data will go into a public repository, okay, at JGI, but you'll have access to that sequencing potential. So it's quite attractive in that way. A lot of things to discuss around the selection of exemplar isolates, what taxa, what species. I think a lot of times as taxonomists we want it to be the type species. A lot of times types are not indicative of the particular clade of interest around a particular industrial or ecological question, or even in the context of phylogenetic diversity. So I think there's some things to think about there. We'll be developing a research coordination network. We've had RCNs in the past. They've been quite successful. Deep Hypha, FESSN. And we haven't decided on the name, but we're thinking the leading name right now is GEM, G-E-M, for Genome-Enabled Mycology. So we'll see. And just a few of the things that we'll be focusing on are phylogenomic analyses, how to scale this up, what would a metagenomic database really look like, and how would you interact with it. Genome-enabled barcoding, we've heard a little bit about that. And then obviously student training. The skill set that you need for this is very different. It's much more quantitative. There's a lot of things that we need to do to get the next generation of students up to speed. So there's some challenges in this. The selection of taxa is not trivial. DNA quantity and quality. You know, for students today, you've kind of been raised in this PCR generation where a little bit of DNA will get you a long way. And you don't necessarily have to be perfect DNA or that clean. You'll PCR your targets out. Right now, the sequencing technologies still require more DNA, microgram quantities, and high quality DNA. It kind of takes us back to pre-PCR days, at least right now. That will certainly change, but where we are right now. And we see a lot, when you're sampling across this diversity, you run into a lot of issues with respect to biochemical problems, different types of polysaccharides, different types of pigments, different types of things that you don't want with your DNA that inhibit downstream enzymatic manipulation of that. And so it's not one DNA extraction. It's solve as you go. There's some challenges when you think about sampling uncultured fungi. Many of these groups are obligate biotrophs. We don't have cultures. Sporocarps are kind of the easiest group to think about, but those turn out to be metagenomic samples. We've done a few sporocarps, a few species from sporocarps. About half the data is symbols, and about half the data is the environment around it. Spores are definitely going to come into play at some point. Single-cell technologies and sequencing of a single cell is developing. There's also some challenges to think about as far as publications. How do you publish 1,000 genomes? I think we should really consider the development of genomic descriptions that are not unlike species descriptions. This has been put forward in the bacterial community with success, but it also changes this kind of balance between a description and something that is more synthetic, and also with respect to the challenge of attributes for young, especially grad students and postdocs. So I'll stop there, and I'll be happy to take any questions.